Okay, bye guys. Thanks for coming to class.
All right, so welcome to the last day of forensics, I guess. I guess we'll just start again, same way that we usually do. Again, you guys can always ask questions in the chat and stuff. Okay, so today we're going to be talking about trace evidence, and basically what trace evidence is that they're like little pieces of evidence. These are often overlooked. So hairs, fibers, soil, building materials, plant material, fingerprints, bodily fluids, etc. It's a quite large topic, but today we're only going to be covering hairs, fibers, and soil. All right, so what is trace evidence? So trace evidence, again, it's like really small pieces of evidence. Okay, so first, I guess we're gonna go over like hair analysis, and this is basically like, animal hair and human hair. So things to look for in hair analysis, that would be color, morphology, or basically what morphology is, like it's relationships, and then the structure of the hair. So before we actually get into actually identifying hair, I would probably like start with some parts of the hair, and basically this is in the very middle. There's like layers, right? So in the very middle there is the medulla. So over here, it's kind of like in this picture, it's kind of like a dotted line. And then there's the cortex, which is like the main part, right? So it's kind of like the largest part. It's labeled here too. This contains pigment granules, ovoid bodies, etc. And what pigment granules are, like, by the name, they're granules of pigment. Ovoid bodies are like large oval structures. They're not always there, but a lot of times they are. And then this last thing, also misspelled, cortical foci, which are little air pockets in the hair. And then lastly, there is the cuticle. And what the cuticle does is that it protects the hair from damage. All right, so let's start with the cuticle. So what this is that it's viewed with a scanning electron microscope, and I'll show some pictures of that later. And this is also the cuticle can also be cast, and it can be viewed under a compound microscope. All right, so identification. So first there's a scale structure of the cuticle, because the cuticle has like, it's kind of different for a lot of organisms. We'll get into that later too. And then there's the medullary index, medulla shape, and the pigments in the hair. All right, so types of cuticles. These include imbricate, spinous, and coronal. So imbricate is kind of like a flat thing. I guess this picture isn't really the best way to describe it, but it's like it looks kind of flat in here. Spinous is over here. I know it says petal-like. In my opinion, it's basically just spiky. And then the coronal, which they look like little crowns stacked on top of each other. And also in these there are like more subgroups, but there's a lot of them, so I don't think we'll be getting into that. Although definitely if we have time, I could do that. But next, there are the cuticles of different animals. So first there's coronal, and these are like small rodents and bats. And as you can see over here, they're kind of like crown-like. It's a small picture, but they look a little bit, they're like all uniform, I guess, and they're all stacked on top of each other. And then there are spinous, and these are like seals and mink. And these are, as you can see, like very spiky. And then lastly, there's imbricate, so human, horse, sheep, etc. And as you can see, this is definitely, in my opinion, a better picture than here because like you can actually see that they're very flat. And a while ago with the electron microscope or the scanning electron microscope, this is a picture with the scanning electron microscope, and I think this is from a stereomicroscope, but we'll get into the different types of microscopy later. So knowing this, we have some practice questions. So again, like usual, I'll give you guys like 2 to 3 minutes per practice question. Also, if you guys are going to answer, you can answer in chat. All right, so it's been 2 minutes, and it's actually B. So by structure, I kind of meant like the strength and like where most of the, I guess, like part of the hair is. And so it was the cortex. So remember, the cortex is the thickest part of the hair. All right, so question 2. Oh, again, like 2 minutes. All right, so it's been like around 2 minutes. So I think most of you guys, if not all, got it correct. So yeah, it was bat. Again, if we look back here, I will go onto the pattern right after I see. Like over here, it's coronal, so that's one thing to look for, and the only answer choice was bat. So um, it was a bat hair. As you can see over here, again, the coronal cuticle. So yeah, the cuticle pattern is also coronal. Like one way you can tell again that they're very uniform, if that makes sense. They're not like they don't really, in my opinion, like imbricate scales look a little bit like mermaid scales in the way that they don't go fully around, while coronal cuticles do. All right, third practice question, same drill, 2 minutes. All right, so it's been like again 2 minutes. Most of you guys got it right. It was B. So again, the cuticle actually provides like kind of protection to the hair. So without it, it would get very damaged. It's not A because it's not the cuticle that's sometimes absent. The medulla is the one that is sometimes absent, and it also would not become healthier because again, it will get damaged. All right, so moving on, we have the medulla. So you can identify um, hairs based on the cuticle, but you can also identify them based on the medulla. So this is kind of like a line running through the cortex in the hair. Uh, it includes the medullary index, and it also has different patterns. So the only one that doesn't really have like a pattern, I would say, is the cortex. All right, so here is the medullary index. I'm not going to make you guys calculate it like I did with RF value last time. So um, this basically this is the measure of the diameter of the medulla um, to like the diameter of the hair shaft. So it's kind of like a ratio. But this is expressed as a fraction. So again, if you guys remember RF factor, the RF factor was represented as like a number. This one's not a number, sorry. Like it wasn't really usually a fraction, while this one is a fraction. And for human hair, this is less than like 1/3. So and then while other animals are greater than 1/3. And what this kind of means is that like the cuticle is like very thin relative to the hair shaft. Although if I did like some math wrong, like please correct me. All right, so types of medulla. So just like the cuticle, the medulla has different types. So we have first continuous, interrupted, fragmented, and absent. So continuous is basically just one long thick line. So as you can see, it goes all over there. So, and then there's interrupted, which looks like kind of your average dashed line. As you can see over here, it's interrupted. And then fragmented is kind of like little bits of the medulla. It's definitely not as consistent as the interrupted, and absent are generally just not there. So continuous are most animals. The one that I can definitely think of right now is a dog, where the a dog hair, where the medulla is very like thick. And then next is interrupted. So these are most animals. While humans, actually the medulla is either fragmented or absent. So it's definitely not as like thick. All right, so this is like again different animals. The dog hair, this one actually isn't very continuous, but if you guys want to see like a better picture, there definitely is one. And as you can see, some identifying factors. All right, so you could say that the cat kind of looks like a ladder. The mouse is some weird like 3-ladder kind of thing. Rabbit looks like a bunch of little whole circle things pieced together, and a deer, it's like rabbit but more condensed. While dog, I guess if you guys want to see a better picture of the dog and the human hair, I definitely could pull up one, but if you guys don't ask for it, I'll just like move on. All right, so one cool thing about looking at a hair under a microscope is that you can actually tell if a hair has been dyed or not. Basically, so regular hair, undyed hair, virgin hair, if you could say that, the pigments aren't as uniform. I don't have a picture of this, but the natural pigments will have a kind of texture. Think about like, I would say coloring with a crayon. And then while a dyed hair will be just like very uniform, very smooth. So another thing that you can tell, this is probably the most obvious one, is that the roots can also tell if a hair has been dyed. Because if you're gonna dye your hair, it's not gonna grow out the color of the hair you dyed. Like if you dye your hair blue, it's not gonna grow out of your head blue. So there will definitely be a difference between the hair that freshly grows out of your scalp that has not been dyed versus the dyed hair. All right, some more practice questions. Or I did not actually make practice questions for these. Oh, okay. So fibers are separated into 3 categories: vegetable, synthetic, and animal. And a vegetable is a base molecule. The base molecule is usually like cellulose or some kind of like thing like that, like structural proteins. Structural. What is it called? Like polymers. Yeah. Synthetic are usually plastic, like polyethylene. I'm not gonna like terephthalate. And then animal is usually some kind of protein. So an example would be wool, which it is keratin. All right, so these can also be tested by microscope analysis, burn testing, and chemical analysis. But today, I think we're going to be focusing on burn testing. All right, so to do this, there's like a burn analysis key. They also come in as charts, but I find that the key is kind of easier to use. So when the fiber is removed from the flame, so basically what you first have to do is light it on fire. Don't commit arson, kids. But first, if it ceases to burn, go to number 2. Again, if you guys don't know how this works, you look at 1A or 1B. If it's like 1A, you would do what it says, and then 1B, go to the next one. And then you kind of choose between those two. So if it ceases to burn, you go to 2. So or like I'll just go like run through. So if it ceases to burn, go to 2. Fibers have the odor of burning hair, or fibers do not smell like hair, you kind of go to 4, or polyester. So some like I would say straight up identifying factors are polyester, it will like this is not on the key, but it will kind of melt. Wool, like cotton, will smell like cotton, definitely I would say would smell like hair, but also does not say that. But wool and silk, as you know, they are from an animal, so it makes sense that they would smell like kind of burning hair. If you guys don't know what burning hair smells like, it's just like I don't know, smells like burning hair. All right, so vegetable fibers and their identifying factors, because again, there are like different groups, and I forgot to put pictures in these, so I'll probably pull up some more pictures on a different screen. But so cotton looks kind of like a twisted ribbon-y kind of thing. So so I did not put pictures, so here is a picture, or I find a good one and then okay, so I cannot find a good one, so that kind of sucks, but okay, there, or no, that's a better picture. Okay, so here is a picture of a if it's not going to copy paste, here's a picture of cotton, and as you can see, it looks kind of like a twisted ribbon-y thing. It looks, I don't know, it looks very twisted. It has like this curl pattern, if I guess you could say that. While linen looks like bamboo-ish. I don't know if you could exactly say that, but it looks, they're like little like indentations on the actual hair. Again, if I can find a good picture, I should probably done this like yesterday, but so I can't find any good pictures, but it looks like bamboo. If I can find a picture, I'll get back to this later. All right, so animal fibers and their identifying factors. So wool, it has an imbricate cuticle again. It looks kind of like the human one. Silk is very smooth, like it's like consistent. Cashmere, the cuticle, the scales are placed very far apart. And Angora, so if you guys don't know what that is, that's like hair, kind of, I think it's a rabbit, and it has a medulla that looks like a cat. So you can see if these are from animals, you can kind of use like hair analysis on it. All right, so synthetic. So these are rayon, acetate, nylon, acrylic, and polyester. In my opinion, they look very synthetic under the microscope. They look like you'll kind of be able to tell just straight up looking at it, just because like they're very consistent and very straight. All right, so if you guys, I did not put in picture, and I found a good picture of like all of them. So again, if you will copy paste. Okay, so over here you can see like wool, it has like an imbricate cuticle. Cashmere, again, spiky-ish, not spiky, but like very far apart placed. Silk, it's very long, smooth, and consistent. Linen, I guess it's also like not, in my opinion, not a very good picture, but I guess we'll have to deal with it. So as you can see, they're kind of like these marks, like indentations. Cotton, this is a much better picture of cotton, where it looks kind of like a twisted ribbon. And again, polyester, you can kind of tell that it's a synthetic because it's very long and very consistent. All right, so after this, we have practice questions again. Sorry for making y'all go through these, but Again, 2 minutes per practice question. By the way, as for formatting this, you can just put like uh vegetable ABC C or something, and then like animal ABC, and then um what's it, synthetic ABC. So if that makes it easier. Do you guys want more time, or do you want me to go now, or like start I guess giving the answer now? All right, so this is kind of I'm gonna try to write out the answer like color coding it, because it is kind of like a sorting. So green is going to be vegetable, red um, animal, and blue synthetic. So wool is going to be animal because it comes from a sheep. And then cotton, since there is a cotton plant, it's vegetable. And then linen is also vegetable, so it's um, green. It's a plant or vegetable fiber. So and then silk is, again, it comes from silkworms, so animal fiber. And polyester is it's like synthesized chemically, so it is synthetic. All right, so I'm gonna clear this, and we can go on to the next question. All right, again, 2 minutes. All right, so it's been like 30, no, sorry, not 30, like 2-3 minutes. So again, if you guys want more time, I could give you guys more time, and then or else I'll just explain it. All right, so nobody's gonna ask for more time. I'll just like put it in. All right, so this is a true or false question. So again, yeah, true or false. And it says the base of for many synthetic fabrics is cellulose, and cellulose is and it's poly. An example of the synthetic fiber is polyester, and cellulose is a polymer. So this is actually false. I would say cellulose comes from plants. So many plant fibers actually come from cellulose, unless I guess this one could be kind of confusing because of like rayon, which is regenerated cellulose, but um the part that says for example a synthetic fiber is polyester and cellulose is a polymer. It's actually the base of polyester actually is not cellulose, it's polyethylene terephthalate, which is a type of plastic. All right, third question, another 2 minutes. All right, so if you don't know, like if you don't really understand the question, I could kind of reword it. So basically, if you see wool and you know it's wool, what would make it like? What would make you think that it is wool? And again, if you guys don't know, always just like you can throw in a guess. All right, so basically this is if you looked at a piece of wool, why like would you think it's wool? So again, if we go back to here again, it said that it has like an inner cuticle. So it has like yeah, over here you can see that it actually has a cuticle, and the cuticles are placed very close together. So that would be like one way to identify the wool. All right, so after this we have soil. These are differentiated based on appearance and color. And basically, as you know, some soil can be darker or lighter than others, and some can also be like some can also be more coarse or like that than others. So these can be described as light, sandy, clay, loamy, and we'll get into that later. And then again, what texture is kind of different from what you would honestly like usually say as it's how much sand, silt, and clay is present in the soil. All right, so this is sand, silt, and clay. They're basically like particles that make up soil. So sand has the largest particle size, silt is in between, and clay is the smallest and most fine. So you want to kind of imagine the size differences. If sand was as large as a basketball, and silt was then silt would be as large as like one particle of sand was as large as a basketball. Then silt would be the size of a baseball, and clay would be like the size of a golf ball. So definitely like there is quite a big difference. All right, so definitely there's more than one type of soil. So if you guys ever walked into Home Depot and bought like a bag of soil, um yeah, that's not the only type of soil that exists. And here's a chart. It's the 6 types of soil. So sand is what you would find on a beach. So it's very gritty. It's mostly, again, sandy particles, and basically it's weathered rock. Then there's clay. This is very smooth because of how small the particle size is, and it's kind of sticky. This can be found in a lot of in a lot of places. And all right, so then there's silt, which is very slippery. I would say like this is kind of sediment and stuff. They're medium sized because again, sand is the largest and clay is in the middle. So clay is the smallest. So silt I guess would kind of feel like an in between of sand and clay, and these are found in like riverbeds. All right, so peat. This is made from decaying organic matter. So this is probably what you find in like a garden or something because of the decaying organic material. And this is very compressible, and this is found in bogs and stuff. And okay, so then there is loam. Loam is the best kind for agriculture, so it's like best for planting and stuff. This is actually, I feel like this one would be found more in gardens. So this is like a mixture of everything. And it's also very like good. It's good for most plants. Then there's chalk. It feels chalky. It's very bad for agriculture. It doesn't retain water, and honestly, just like in my opinion, not very good. So, and then you might be wondering, what is this giant triangle over here? So this giant triangle is kind of another way to help identify soil types. So there's different sections, and this is kind of like I guess a three-way kind of, if you guys ever learned to plot points, it's kind of like this but with 3 sides. You have the clay percentage, the silt percentage, and sand percentage. So I think the you would find like the clay and then go somewhere, I guess like say here, and then silt, look in that area, and then like sand percent. So this is just another way to identify it. All right, so more practice questions. Yay. This again, 2 minutes per cross practice question, and then I'll explain it. Also, yeah, just type your answers in chat. All right, so it's been like 2 minutes. It's it's actually A. So the largest particle is A. It's sand. So as you can remember, like see that sand is like the largest one because clay is very like soft, very I don't know, like and then silt is kind of an in between. Again, sand, if sand were the size of a basketball, then silt would be the size of a, I think, baseball, and then clay would be the size of a golf ball. So sand is definitely the largest particle. All right, to practice question 2. So this is kind of like the opposite of practice question 1. Technically I already did give the answer, but yeah, again, 2 questions. Sorry, 2 minutes. All right. So like we or I think there's yeah, so what all of you guys got it correct. It is clay. I don't I think I don't really know if I need to explain this one because it's practically the same explanation in question 2. And question 3 again, 2 minutes. All right. I see that, I don't know if y'all can, okay. It is C for the reason that there are so many C's. I don't know. I feel like C is a very good letter. Yeah, it is C because if you look back, okay, here it says that loam is the best type of soil for agriculture. And as for the rest of them not being soil, technically you can kind of like also agriculture is plants, so loam would technically be best. As for other kinds not being soil, technically you can kind of think of them as soil. They definitely do fall under the category. All right, so if we're talking about all of this stuff, you have to view it, right? So you have so definitely we have the electron scanning microscope. So these are like the types of, okay, that should say microscopes used in microscopy, not microscopy used in microscopy, but we can just ignore that. So there's three kinds that are generally used. There's the electron scanning microscope, compound microscope, and the stereomicroscope. All right, so first we have the stereomicroscope. This is, in my opinion, the worst kind. It's very boring, can't zoom in a lot, but it does allow for 3D images. So I guess also the kind of boring part is my opinion. Like, don't take that actually as like part, but these are generally used for hair and fiber analysis because of being able to see the actual 3D image shape. All right, so next we have the compound microscope. So in my opinion, these are much better than a stereo microscope. Yeah, I particularly do not like stereo microscopes. These are much better than stereo microscopes. Generally because you can actually zoom in a lot more. So as you can see, there's a picture of some, I think, microorganisms that you definitely like see, like you have to see the whole like, I don't know what this is, insect in this picture, right? So you can zoom in a lot more. These will detect minerals and metals in like soil and stuff, and these are also used for blood analysis. All right, so next one is the best one, in my opinion. Always, this is my opinion, not actual facts. Well, actually, but anyways, these are, as you can see, here is a picture of a bug. I'm sorry if that scared anybody. There's a really high magnification for these, and it produces a pretty clear 3D image of it. And like this one can zoom in a lot. Definitely, like, you can't really tell for this picture, but it can zoom in a lot. These are usually mainly used to view the cuticle, and there's not really color. I mean, I guess you could add color to it, but the original picture will not have that color. Again, I think I can show a picture of if I scroll back like go really far, there, I think here, okay, not here. Oh, here. This is, I think, a picture with the scanning electron microscope. All right, back to here. All right. Next, we have the compound microscope. So how does this actually work? This does require lots of physics, and I personally don't like physics, so I'll just kind of go over this. These have two sets of lenses, and they cause light to bend towards each other, and they form like a cone, and this will go like over and over that, and will like cause an image to appear like a lot bigger. I can't answer many questions on this because I don't particularly like physics, and yeah. All right, so stereo microscope, the boring one. Unlike a compound microscope, this actually uses two naturally reflected off two like paths reflected, and this unlike a compound microscope, like over here there's a light. The biggest microscope, no idea. Out of these three though, the biggest microscope, oh, there's a question in the chat. I don't know if you guys can all see it because it is only to hosts and panelists, but it asked what the biggest microscope was in the world, and I can't answer that, but the biggest out of these three is definitely the electron microscope. It's all fancy and stuff. This is unlike a compound microscope, it actually uses two light paths, like, you know, over here, two eyepieces, and it uses light naturally reflected off of the specimen. Oh, and because they're using the light like naturally reflected off of it, it will not, you can't really go as, like, can't zoom in as much as you can with the compound microscope. All right, how does the scanning electron microscope work? This will scan the surface of the specimen with a beam of electrons, so this time not light. The electrons will pass through a series of like lenses and stuff. So as you can see over here, there's like lenses and fields and stuff, and it will create an image displayed on the screen. So over here, as you can see, there is the compound microscope and the screen. All right, so went over this pretty quickly. So then we have practice questions, and afterwards there is a survey. I think you guys probably had to do this for the other classes, but you know, Rich told me to do it, so might as well, right? So again, like I would say actually, because I have to give you guys a survey. I'll give you like 1 minute on this. Also, there's like I think a fire truck outside my house, so if you guys can hear that, sorry. All right, so the reason that the stereomicroscope can't zoom in a lot is again, like I mentioned before, it only uses like light naturally reflected off of the specimen, so you can't really zoom in as much as you can with light actually added there. All right, question 2, again, 1 minute. All right, so my timers went off. So these are the electron scanning microscope and the and the what's it called? Okay, I momentarily had a brain fart, but and the stereo microscope, because these you can actually see in 3D. All right, so thank you guys, and actually there is a survey so that I have to like give you guys. So I guess I'll give you guys like the rest of the class to fill in this survey. You can find it on the KTK website. It's also on I'll also send the link in the chat. But if you guys miss it, you can always go to the KTK website. Or if this doesn't work, I would say, just then go to the website.